We are fortunate to have today's lecture by Dr. Jan Beumer. Jan is Associate Professor of Pharmaceutical Science at the University of Pittsburgh and the Director of the University of Pittsburgh Cancer Institute's Clinical Pharmacology Analytical Facility. Jan earned his Doctorate of Pharmacy degree and his PhD from Utrecht University in the Netherlands. He is a certified clinical pharmacologist through the Dutch Society of Clinical Pharmacology and Biopharmacy, and a diplomat of the American Board of Toxicology. Jan is an expert in preclinical and clinical metabolic studies with a focus on mass balance studies. His research interest surrounds the translational pharmacology of anti-cancer drugs, and he has published over 100 manuscripts in peer-reviewed journals. I'm confident you will enjoy today's lecture.
Hello there. I am Jan Beumer. I am an Associate Professor of Pharmaceutical Sciences and Medicine at the University of Pittsburgh. I will talk to you about drug absorption and bioavailability. This is the overview of my talk. I will introduce some PK/PD and definitions. I will discuss the physics and physiology of absorption, focused mostly on oral absorption. I will discuss the biopharmaceutics classification system, factors affecting oral absorption. Then I will discuss flip-flop kinetics, other extravascular doses, routes, and I will finish up with bioequivalence. So this is a slide that really shows the basis of pharmacology. On the left you see the pharmacokinetics, what the body does to the drug, and on the right you see the pharmacodynamics, what the drug does to the body. On the bottom left you see a graph of an intravenous concentration versus time profile. There is an instantaneous distribution followed by a first order elimination. Of course, when there's an absorption step involved, this changes. The concentration starts at 0, and at a certain rate the drug gets absorbed, presents itself to the systemic circulation, reaches a peak, and is then followed by this elimination phase. On the right hand we see a classical dose-response curve where the exposure is expressed as a log value versus the effect following the Emax model. Of course, we got to keep in mind that there's not just one dose response, there are really two dose responses. In the green we see here the desired dose response curve for the intended effect, and in the red we see the side effect dose response curve. And the gap in between these two curves is really the therapeutic window that we are targeting with our therapies. Another consideration is that there are likely multiple red dose-response curves because every side effect has its own dose-response curve. If we now take these two graphs and put them together conceptually, then we see here in green the minimally effective concentration, in red the minimum toxic concentration, and as the drug gets absorbed, it crosses the minimum effective concentration and the effect has an onset, it starts. It has a maximum effect, the elimination takes place, and when the concentration drops below the minimum effective concentration, the duration of the effect has ended. And this again is the therapeutic window that we are targeting between the red and the green. And if absorption changes within a patient, between patients, we can reach high concentrations resulting in toxicity, or lower concentrations resulting in lack of efficacy. So I just want to present some definitions of absorption and bioavailability, what this talk is about. These are definitions from Goodman and Gilman. The absorption is the movement of a drug from its site of administration into the bloodstream or central compartment and the extent to which this occurs. Bioavailability is defined as the fractional extent that drug reaches its site of action or a biological fluid. And here you can already sense that these definitions are not extremely specific. There is sort of a quality to it. It's the bloodstream, the central compartment, the site of action. It's not very defined. Another definition of bioavailability that I do want to share is the one in the Code of Federal Regulations, because this is the definition that the FDA uses. It's defined as the rate and extent that an active moiety is absorbed from a drug product and becomes available at the site of action. So this bioavailability, this fraction that gets absorbed, is really based on the area under the concentration time profile. And so here on the right we see the area under the concentration time profile, the real surface area under this curve when concentration is plotted linearly. And so one determines this area after oral administration in green here, and after IV administration, the curve in red. And in this case, the areas under both curves are identical, and so the fraction absorbed is 1. On a log scale, this results in these plots with parallel terminal elimination phases. If the fraction is less than 1, then the oral curve will drop down correspondingly, and you get a lower exposure. So in formulas, this is depicted as down here, where the area under the concentration time curve after IV dosing is the dose divided by the clearance. The dose we can determine by choosing the dose, and the clearance is a biological parameter that may differ between patients. If the dose is given extravascularly, then there is a fraction that gets absorbed, right? This bioavailability, and so that then enters the equation. And F can be isolated, as here, and so you can determine experimentally this F by ratioing the dose-normalized AUC after extravascular administration divided by the dose-normalized AUC after IV administration. Next, physics and physiology. So here we see for oral administration the whole path from disintegration of a solid dosing form to individual particles in the gut lumen, to dissolution of individual molecules, diffusion towards the gut wall, through the gut wall, entering into the portal vein, then reaching the liver, and then finally reaching the systemic circulation. And at any of these steps, there is the potential for loss, as depicted here. Now, absorption is commonly considered to take place between the dosing form and reaching the portal vein. But for oral bioavailability, it also includes this step of getting past the liver. The first time that each drug molecule has to make it past the liver. And so this is called the first pass effect. So just focusing on some of these processes individually. The disintegration of a solid dosing form is rate limited by the liquid penetration into that dosing form. And so drug release will be affected by a variety of factors. The excipients that are chosen, often fillers, or compounds that aid in the processing of the dosing form. The tablet compression strength is important for this liquid penetration. And of course, any coating or matrix that is added to the dosing form to affect these processes. So if you embed the active pharmaceutical ingredient, the API, in a polymer matrix that dissolves or swells at a slower rate than the drug, then this will impact the process of liquid penetration. It also can increase the disintegration by swelling up and breaking up the solid dosing form. In addition, if you coat the tablet, you can create a mass transfer limiting barrier, and so you can create a prolonged exposure, an extended release form, or you can make this barrier soluble based on pH. And so this is how you can create enteric coated tablets that do not dissolve in the stomach juice under acidic condition, but do dissolve once the dosing form hits the intestines. Dissolution is described by the Nernst-Brunner equation as depicted here. The change of the concentration in a solution is dependent on the diffusion coefficient, the surface area of your particles, diffusion layer thickness, medium volume, and solubility. And so if we look at the baseline curve, the starting point, this black line of a base, bases are usually not very soluble, and so the solubility is very low, and also the kinetics are very low. And so you reach a solubility with a very slow pace. If you now take this same chemical compound, the base, formulated as the base, but you you decrease the particle size and thereby increase the surface area that is available, then you will get to the same final concentration, but you'll get there quicker. And so this is the difference between kinetics and dynamics. Thermodynamically, the solubility hasn't changed, but kinetically, you get there sooner. Other ways of impacting the dissolution process is to formulate a compound as a salt. This increases the solubility, so you get to higher total concentrations, and of course the rate therefore increases as well. If you note here, the change in concentration, this rate, is directly proportional to solubility. Higher solubility, you get there quicker as well. In addition, you can also formulate compounds as an amorphous compound, different polymorphisms, hydrates, anhydrates, etc. And the way you can think about this is that if a solid is amorphous, then it's not in a stable crystal structure, and so it requires less energy for a molecule to leave that solid form and enter the solution. And so solubility is enhanced. These different dissolution profiles will then result in different concentration time profiles inside the body, and that is depicted here. So the next step is to transport these molecules across the enterocyte membrane and enter the biological system. And this is depicted here, the different ways that can happen. Passive diffusion, transcytotic vesicles, we'll not go into that too much. Passive diffusion through the paracellular route with molecules that are hydrophilic and small. Facilitated diffusion with carriers, we'll go into that quite extensively. And active transporters, which include the solute carrier family and the 8B binding cassette family. We'll also discuss these. So passive membrane diffusion follows Fick's first law, here depicted on the right. And so the flux is the diffusivity times the concentration gradient. And it is the concentration gradient across the membrane. And so if you then rearrange this formula, what you get is diffusivity times the difference in concentration at the beginning and the end, divided by membrane thickness, which would be a constant. Now here we have the concentration, this is a y-axis, the concentration in the solution, which you reach by the dissolution process that we just discussed. And then in the membrane, the concentration jumps. And so this difference is the partition coefficient of your molecule into the membrane. And this is Kd. The same happens on the other side. There is this ratio of concentrations in membrane and solution, and this again is the Kd. Usually one considers this concentration to be a sink condition, so it's 0. And so the concentration gradient is linearly related with the concentration at the beginning of the membrane, which through the partition coefficient is linearly related with the solubility. And so if you now combine this diffusivity, the partition coefficient, you get the effective permeability. And this is a parameter that is often determined with experiments. And so as you now can imagine, the diffusion through a membrane is very much dependent on the partition coefficient, how much does a molecule like to go into the membranes, and the solubility. Now of course, this doesn't take into account that molecules are not always in the neutral form. Only the neutral form passes membranes, but their drugs are often acids or bases. And so I'll discuss this in this slide. This partition, this acid-base equilibrium is described by the Henderson-Hasselbalch equation, depicted here top right, and the ratio of the two forms, the chemical forms that occur, is described here, 10 to the power pH minus pKa. Now I just mentioned that only the neutral form will pass membranes, and as we see here, for each one neutral molecule, there are 1,000 anions, and this is based on the pH in plasma, 7.4, and the pKa of this compound, 4.4. The total number of molecules is depicted here, 1,001. In the gastric acid, with a pH of 1.4, this equilibrium is the other way around. For each one molecule of neutral acid, there are 0.001 molecules of anion. And so as you can see here, the equilibrium in the acid is pushed to the neutral form, and on the plasma side it is taken away from the acid, from the neutral form. And so this really favors absorption through the lipid membrane. Now still, weak acids are not absorbed in the stomach very much. And this has to do with the membrane thickness in the stomach and also the limited availability of surface area. Here we have the same situation for the base. I will not go through it in detail, you can do that yourself, but suffice to say that the ratio is completely different. It's 1 to 1,000,000. And so weak bases are very disfavored for absorption from the stomach, understandably so. Now this is not necessarily a bad thing, because what it does favor is the dissolution of the compound. As you can see, if a molecule is dissolved from its solid form, it immediately gets ionized, and so this drives the equilibrium to dissolution. And then once it's all in solution and these molecules get dumped into the intestine with a large surface area and a neutral pH, then absorption is favored at that point. Here we can see a bit of a bigger picture. So here's the intestinal lumen. Here's the enterocyte. Molecules enter through these carriers. They may be effluxed by PGP. And what happens is that they get repeatedly exposed to CYP3A. So molecules enter, they may be metabolized, and then they may be pumped back. Some molecules may make it past the enterocyte and enter the portal vein. And here, these pumps are no longer in the same membrane and in opposite directions. Once a molecule is in the portal vein, it may be facilitated into the hepatocyte. Again, there's a likelihood of being metabolized. And even if it doesn't get metabolized, it may be pumped out into the external environment through bile and forming the enterohepatic recycling pathway. And so this system is very nicely set up to prevent xenobiotics from actually reaching the systemic circulation. Here's a depiction of the enterocyte with the variety of pumps that have been characterized. And I will not go into this in much detail. There are other lectures that will address this. But suffice to say that drug-drug interactions and polymorphisms have been characterized and are being discovered every time. So this is the metabolism that takes place in the enterocyte. I just mentioned CYP3A, and this is indeed 80% of the phase 1 metabolism that takes place in the enterocyte. There are also phase 2 metabolic enzymes, UGT1A being the predominant family. In addition, there is gut flora, the bacteria in the gut lumen, the microbiome, and these can hydrolyze and reduce and change the activity of compounds. In addition, this gut flora has been shown in animals to modulate the gut and liver activity of CYP and phase 2 enzymes. And with the increasing study of the microbiome, this is not a surprise. So here are some examples of hydrolysis of lovastatin into its active compound, inactivation of digoxin, through reduction, release of the active components of sulfasalazine and prontosil, and this is the reaction of nitrazepam to its amino metabolite. Now the intermediate step is not depicted here, but that is a hydroxylamine, and that is a potentially carcinogenic component. So here I just want to provide another picture of this interplay of phase 2 and phase 3 metabolism. Phase 3 metabolism is often defined as these carriers and transporter effects. And so here you see a molecule repeatedly being pumped out and exposed to CYP3A, increasing the likelihood of metabolism and reducing the likelihood of an intact molecule, xenobiotic, actually reaching the systemic circulation. Here we show the hepatocyte with all its pumps and also the biliary canaliculi. And again, these hopefully will be discussed in other lectures, and there may be drug-drug interactions and polymorphisms that play a role here. So the last step before a molecule can reach the systemic circulation is metabolism in the liver. And so that is a big component of the oral bioavailability. The total bioavailability is often split up in the fraction of the dose absorbed by the intestinal epithelium, the fraction escaping gut metabolism and entering the portal vein, and then this last factor is the fraction escaping hepatic first pass extraction and entering the systemic circulation. So far I just discussed very simplified diagrams, but of course the GI tract is very diverse. And so some of that is depicted here, where the pH changes as you go along the GI tract, and especially the surface area differs vastly. So the factors impacting absorption are the surface area, vascularity, pH, fluid volumes, presence of other substances such as bile, and GI motility and emptying. And here on the right we see why these small intestines have such a small surface area. There are villi and microvilli that really increase the surface area for absorption. So here I put that in a table, and so the salient details to look at are that esophagus is not really a place for absorption. The mouth is, potentially. Jejunum and ileum have a large surface area. And the rectum and the mouth may actually avoid the first pass effect. The mouth and part of the rectum blood flow bypasses the portal vein. And so, for example, there are oral tablets that will dissolve within a minute in the mouth and release its compound to be taken up very quickly, such as nitroglycerin tablets. Now the pumps and the enzymes that I discussed, they are not expressed to the same extent across the GI tract, and that is depicted here. So physiological factors that cause poor bioavailability are listed here: diseases of the gut, functional integrity, insufficient time for absorption, food effects, drug complexation, degradation, poor dissolution or permeability, transporter saturation, efflux pump substrates, gut metabolism, and hepatic metabolism. And some of these items I will touch upon as we go along. Another aspect is saturability of absorption. It's one of the points in the previous slide, and here illustrated by nilotinib. So as nilotinib, the daily dose, single dose, is increased, the exposure, as expressed by the AUC over 0 to 24 hours, increases pretty much linearly. But starting at about 400 milligram, it plateaus out. More drug just doesn't get in. However, if you split this once a day 800 milligram dose in twice a day 400 milligram, the exposure over that 24 hours will increase 35%. And so a major PK objective in phase 1 trials of oral drugs is to document the potential presence of PK futility. If you go up in dose and you don't get more absorption. So now we'll discuss the biopharmaceutics classification system. This is a graph that underlays the development of this system. And so here we see the human permeability over a jejunal membrane and the fraction absorbed in humans. And as you can see, that is a pretty nice relationship. And so the thought behind this system is that dissolution and GI permeability are the fundamental parameters controlling the rate and extent of drug absorption. And when in vivo dissolution is rapid in relation to gastric emptying, the rate and extent of drug absorption is unlikely to be dependent on drug solution and/or GI transit time. And so we now get this system where drugs are either high solubility or low solubility and high or low permeability, resulting in class 1, 2, 3, and 4. So how is this defined? Solubility is high if the highest strength of a drug is soluble in 250 ml of aqueous media. Permeability is measured by the rate of mass transfer across human intestinal membranes, or documented in humans. So the purpose of this system, the BCS classification has been extended to the biopharmaceutical drug disposition classification system, BDDCS. The purpose is to predict drug disposition and potential interactions. It is based on the same parameters, and the thought behind it is that if a drug passes membranes easily, then if it gets filtered by the kidneys, it may easily be reabsorbed from the renal tubules. If a drug easily gets, if a drug gets excreted into the bile and it passes membranes easily, it may easily reabsorb from the bile canaliculi. And so, just like I showed the interplay of pumps and CYP3A in the enterocyte, this results in the cycling of a drug repeatedly through enterocytes and, through the liver, and predisposes a compound to metabolism. So high permeability compounds are likely to be cleared metabolically. And so this is how this is expressed. Again, this is the original BCS classification system, and now applied to clearance. Class 1 and 2 are often metabolized, and class 3 and class 4 are usually not metabolized and excreted by the kidney or the bile, unchanged, because they are low permeable and they will not be reabsorbed. They may be excreted mostly unchanged. Now I will discuss factors affecting oral absorption in more detail. So food may impact the rate and extent of absorption. These are some of the reasons why. Food can physically or chemically interact. It slows gastric emptying, prolongs transit time, raises pH, increases bile output and motility and blood flow. It may change the luminal metabolism, and bile and lipid components will inhibit transporters. The effect is, of course, dependent on the meal size and composition. Another pattern that I would like to present to you is the pattern depicted here. And it is the variability of absorption plotted against the extent of absorption. And so if a drug is extensively absorbed, then the variability is lower. And you can imagine this. If a drug is 90% absorbed, a couple of percentages more or less is not going to be a big relative difference. However, if a drug is only absorbed 1 or 2%, then 1 or 2% more or less is a relative big difference. So extending the BDCSS classification system to this food effect, fatty meals. So for a class 1 compound, the extent of absorption doesn't change much, but because gastric emptying is slowed, the maximum concentration will often be increased. Class 2 compounds, the extent will go up. These are low soluble, and what happens is compounds are often solubilized more by food and the bile salts that are released because of food that are triggered. This will increase the solubility of these compounds, and the food components will inhibit efflux pumps, and so this will also increase the extent of absorption. Class 3 compounds are often dependent on transporters to gain entry into the enterocyte, and these carriers can also be inhibited by food components like bile and lipids, and so the extent is often decreased. Class 4 compounds are always complex to predict because all these effects can have an effect. So here's a pertinent example of a food effect. Here we see the average lapatinib concentration fasted with a low-calorie, low-fat breakfast and with a high-fat breakfast. And so the food effect on average is about a factor 4. But if you look at individual patients depicted here, some of these patients have a 10-fold increase in absorption with a high-fat breakfast. The idea behind this is that food generates bile release, chylomicrons are formed, and this drug, lapatinib, very lipophilic, dissolves in the chylomicrons, and these are taken up by the lymph system. And the lymph system bypasses the portal vein, bypasses the liver, and the lymph gets dumped into the superior vena cava. So there's no first pass effect. Now this is very relevant because lapatinib has a black box warning warning for hepatotoxicity and potentially death. And so high concentrations are potentially dangerous. So currently lapatinib is labeled to be used fasted at 1,250 milligram every day, and it costs about $7,000 per month. The food effect is a 4-fold increase. And so what is preventing people from taking a quarter of the dose with a fat meal? And this has been discussed as the value meal in the references provided here. Additional food effects, complexation and stability. 6-mercaptopurine is inactivated by xanthine oxidase. Milk has a high level of this enzyme, and the activity of this enzyme is not diminished by pasteurization or gastric acid juices. And so this underpins the interaction there. Tetracyclines often complex with metal ions, magnesium, iron, calcium, and so this prevents absorption of this class of antibiotics. And finally, fluoroquinolones also complex with these metal ions, but in addition, recent publications show that absorption of the fluoroquinolones on protein surface of milk actually is a bigger effect than complexation with these metal ions. So the FDA developed a guidance about food effects in 2002, and all oral products need to be tested for a food effect. So they stipulated how this would be designed. It needs to be sufficiently powered, it needs to be studied at the highest dose, and the conditions should be such that you expect the greatest effect on the GI physiology. And so even the meal has been described pretty clearly. The design in general for the fasted piece is an overnight fast for at least 10 hours, dosing the highest dose with 240 mL of water, and no food until 4 hours post-dose, and no water until 1 hour post-dose. The fat component is the same, except that a meal needs to be consumed within 0 to 30 minutes prior to the dose. PK parameters that are documented are listed here, and the absence of a food effect can be concluded if the 90% confidence interval for the ratio of the population geometric means between fed and fasted after log transformation is contained in the equivalence limits of 80 to 125% for both AUC and Cmax. And this criterion comes back in the bioequivalence section of this talk. Next, I want to discuss the flavonoids. And this is an interesting landmark paper where the effect of grapefruit juice is documented on phenylidipine PK. So this study documented colon and intestinal levels of CYP and PGP through biopsies. PK was documented and phenylidipine is completely absorbed, but the bioavailability is only 16%, indicating that metabolism is a big component of the bioavailability. It also studied liver CYP3A4 activity with the erythromycin breath test. No effect was documented of grapefruit juice on liver CYP3A4 activity, CYP3A5 levels, or small intestinal PGP, CYP1A1 or 2D6 levels. The effect was really focused on the small intestinal epithelial CYP3A4 levels, 62% decrease, and this is shown here in the top right graph. There was no change in CYP3A4 mRNA levels, and so this suggests a direct effect on the protein. A correlation was documented between enterocyte CYP3A4 levels before grapefruit juice and the extent of the effect of grapefruit. And you can imagine this. If a patient has a high level of CYP3A4, then there is a lot of CYP3A4 to inactivate by grapefruit juice. And so the change in Cmax is then extensive as well. And so here we can see this effect where with water, the phenylidipine PK, the exposure is low. Together with the first grapefruit juice intake, it goes up. And after the 16th dose of grapefruit juice, it's even higher. And so later on, it was shown that bergamottin and its dihydroxy metabolite actually are responsible for mechanism-based inactivation of CYP3A4. So covalent binding of this compound to the CYP3A4 essentially killing it and preventing it from functioning further. Grapefruit juice flavonoids can also inhibit PGP, and this is documented here with the Keiko2 cell line. Keiko2 cells are grown here on a semi-permeable membrane. They form a nice layer, and drug is applied on one side, and permeability to the other side is documented. And so here we see the control, and after addition of grapefruit juice, the permeability increases. And so this was concluded to be the result of PGP inhibition by grapefruit juice components. So grapefruit juice is not the only fruit juice that causes interactions. Fexofenadine, which is a BCS class 3 drug with negligible human metabolism, was studied after water, the top curve, 25% grapefruit juice, so diluted, full-strength grapefruit juice, orange juice, and apple juice. And so all these juices quite extensively decrease the absorption of fexofenadine. So this is the opposite effect of what we just saw with grapefruit juice and felodipine. And so this is because of inhibition of the influx carrier, OATP, by components in these juices. So now we have these two opposing effects. We have an effect on the influx carrier OATP, and we also have the effect on CYP3A4 by grapefruit juice. And so we have here two probes, ciliprolol and midazolam, after oral administration, and this is with the first dose of grapefruit juice. So there is an effect of of grapefruit juice, and here midazolam, an effect as well. So both OATP and CYP3A4 are impacted. But on day 3 and day 7, we can see that the effect on OATP disappears pretty quickly, whereas the impact on CYP3A4 is more lasting. And so the components in grapefruit juice that inhibit OATP, that is reversible, whereas the impact on CYP3A4 is more lasting. And as we just have seen from bergamottin, from the bergamottin slide, this is a covalent interaction, and so CYP3A4 needs to be resynthesized before it is back to baseline. Here's just an overview of different compounds, different juices, different strengths, different volumes administered. And so every substrate and every juice can have a different extent of effect on the AUC ratio, here depicted below. So flavonoid effects depend on which substrate is studied, what flavonoid is present, combinations thereof, concentrations of flavonoids, and we have to remember that these are natural products, right, food products. And so location, geography of growing them and harvesting time can all have impacts on the relative levels of these flavonoids. The Western diet contains about 200 milligrams of these flavonoids a day, and some very common ones are listed here: naringin, hesperidin, the furanocoumarins, which includes bergamottin, and the last one I want to discuss is hyperforin, which occurs in St. John's wort. Hyperforin is a CYP3A4 and PGP inducer, and when given with imatinib, it decreases the AUC and Cmax of this anti-cancer drug. St. John's wort is a herbal remedy for depression, and so you can imagine that cancer patients may be depressed and take this as a herbal remedy, but in treating their depression, they may actually compromise their anti-cancer therapy. So these transporter effects that we just discussed, they have also been expressed in the BDCSS system as follows. If you have a class 1 compound, then there are no transporters needed to get into the enterocytes, and they enter at such a high rate that efflux pumps will not have an impact. So transporter effects are minimal. Class 2 compounds, they will enter membranes very easily, but not to a high extent because their solubility is low. And so efflux pumps may impact that absorption process. Class 3 compounds, they don't enter membranes very easily, and so they need the absorptive transporters. And so interactions at that level are likely. With class 4 compounds, all transporters could have an impact, and it's hard to predict which effect predominates. Next, I want to illustrate the effect of gastric pH using dasatinib. The solubility of dasatinib at pH 2.6 is 18 milligram per ml, whereas at neutral pH it's almost 20,000 times less. And so if you take dasatinib with an H2 antagonist, which increases the pH of the stomach, then the absorption will be less. And so depending on the tyrosine kinase inhibitor studied, and depending on the specific antacid used, these effects can exist or not. And for some drugs, it's not studied very well, and so the impact is unknown. The opposite effect can also happen. Acidity can cause degradation of a compound, such as in the case of alendronate. And so for this compound, the AUC is increased by a factor of 2 in the presence of ranitidine infusion, which increases the gastric pH to approximately neutral levels. Here is an overview of tyrosine kinase inhibitors and food effects, antacid effects, and what advice is given in the package insert, in the labeling, without food, with food, and avoiding antacids or not. Next, I want to discuss altered anatomy. We shouldn't assume that patients have an intact GI tract. People have Whipple procedures or gastrectomies because of cancer, or gastric bypass because of obesity. This is an ever-increasing percentage of our population, and these surgeries happen ever more frequent. This causes reduced gastric volume, which may increase the toxicity of certain compounds because of a smaller volume, same dose, higher concentration, increased pH, changes in gastric emptying, and increased motility. Bile salts, which are dependent on enterohepatic recycling to be taken up and reused again, that process gets interrupted and can impact the absorption of certain drugs. And the duodenum gets bypassed, and so reduced surface is available for absorption. And as you can imagine, extended release formulations in this setting are not appropriate. So the different surgeries that exist, because there are different types of gastric of bypasses, they can be either focused on malabsorption, gastric restriction, combinations thereof, or surgeries that combine maldigestion with malabsorption and gastric resection. The most popular ones at this point in time are the sleeve gastrectomy and the Roux-en-Y gastric bypass. And here's just an illustration of the variety of different surgical techniques that have been applied. And here I will illustrate the different impacts that can have. So in this paper, and there are not a lot of good pharmacology studies that are being done in this population, but this is an example where there's the Billroth 1 procedure, the Billroth 2 procedure, and the Roux-en-Y procedure. And so for one drug, here in the dotted line we see the healthy volunteer control group, and the absorption in all these gastric bypass patients has been decreased. But for another drug, the impact is different. Earlier Tmaxs, later Tmaxs, and so depending on the surgery that has been performed in the individual patient and the drug that is being taken, the effect is really hard to predict. This is 2 examples from my own lab where we studied temozolomide PK after Roux-en-Y bypass with no apparent change in PK compared to literature values. And below is a graph of imatinib PK before, in black, and 3 times after sleeve gastrectomy. And you see about a 50% reduction in plasma concentrations. And the maintenance of response for imatinib to control CML or GIST has been related to trough concentrations. And so this is a clinically very relevant effect. As you can imagine, industry has been looking to select chemicals for drug development based on their bioavailability. And so how can you predict that? How can you select out of your hundreds of compounds the molecule that is most likely giving you good oral bioavailability? As you can imagine, with models in general, the complexer the model, the less it is suitable for high throughput. And so the complexity of the absorption process makes it impossible for models to be relevant and simple. So there are three items that I want to briefly discuss. First, the log P. This is very closely related to the KD that we discussed in the membrane diffusion slide. And so this is really the partition coefficient between octanol and water for a drug. Now, why are we using octanol? It's because the polarity of octanol is very similar to that of membranes, of lipid membranes. And so this is a good approximation that is easily obtained in vitro. Next, there's the Lipinski rule of 5 and the Keiko-2 cell line system. And so these are systems that are models that are used by pharma to select lead compounds. So the Lipinski rule of 5 is listed here. Basically, based on a big data set, poor oral absorption is more likely when there's more than 5 hydrogen bond donors. The molecular weight is more than 500. The calculated log P is more than 5. Too much of a good thing is not great either, because then compounds won't leave the membrane. And more than 10 hydrogen donor acceptors, bond acceptors. Exceptions to the rules are often transporter substrates, as you can imagine. Other people have similarly come up with rules: ionization at intestinal pH, particle size, stable polymorphs, low aqueous solubility, molecular flexibility, and polar surface areas. The Keiko-2 cell line we've discussed briefly before. This is derived from a human colorectal carcinoma cell line. It is cultivated for about 3 weeks, and it consists of a polarized monolayer with a brush border, microvilli, and tight junctions. And so you apply the drug on the top side, the apical side, and you monitor how much shows up in the basolateral side. This is some of the data associated with the Keiko cell line. So here we see the apparent permeability, these top two graphs, plotted against absorption in humans. And so as you can see, the relationship is pretty decent. Here we see a different graph where the Keiko permeability is plotted against the human jejunum permeability that is part of the BCS classification. And again, a reasonable correlation. So like any model, it's not perfect, but it is a tool. The disadvantages are the long differentiation period, the passage number and inter- and intra-laboratory variability, the absence of a mucus layer, the absence of a first pass effect, and the fact that paracellular transport and CYP expression is lower in the Keiko-2 cell line than in vivo. And this has resulted in the generation of subclones with different levels of transporters and enzyme expressions. Now I will discuss flip-flop kinetics. So here we see the process of dissolution, absorption, and elimination. And each of these processes has a rate constant. For simplicity, further I will sort of combine the dissolution and absorption rate. There's an absorption rate and an elimination rate. So here in this table there are 3 scenarios, and in each of these scenarios there is one rate constant that has the smallest value, and that then is the rate limiting step. As a rule, absorption rates are often much higher in value than the elimination rate. And so the elimination rate is rate limiting. But exceptions exist. So here in a graph, we see in green the common situation, where Ka is higher than K elimination. And so we are accustomed to thinking of the terminal elimination phase as reflective of the elimination process. But here I simulated a curve in blue where we reversed these values. And even though the height of this curve is a little different, the fraction absorbed changes if you change these parameters, the slope of this terminal phase is identical. And so what we need to understand is that the terminal phase of a concentration time profile is reflective of the rate limiting step, which not necessarily is the elimination rate. So how can you find out whether you have flip-flop kinetics, where these rates are are reversed? Well, you need to do an IV study. And so these are the two situations. If you don't have flip-flop kinetics, the common rule, your IV curve and your oral curve will have parallel terminal phases. The elimination rate is rate limiting. If they are not parallel, then you are dealing with flip-flop kinetics. And in this case, your apparent terminal half-life in your oral curve is reflective of your absorption rate. And actually the upfront piece, that is your elimination rate. This is a real example from my laboratory in mice. So here we have the IV concentration time profile as a solution, of course. The oral concentration time profile, again, as a solution. And then we performed a study orally with a suspension in 1% carboxymethylcellulose. This is a thickening agent to stabilize the suspension. And what we see here is characteristic of flip-flop kinetics. And so what has happened here? Well, it turns out that this thickening agent also reduces the diffusion rate. And so in the gut lumen, the drug reaches the membrane at a much slower rate. And so diffusion here is determining this terminal phase. It has become the rate limiting step. This effect can be put to good use. This is not oral absorption, this is absorption from a patch through the skin. And so instead of giving daily doses with a highly variable concentration time profile, you can apply one skin patch where the release from the patch or the skin absorption is rate limiting. And so you get this much more prolonged concentration time profile where your average concentration is maintained much longer with one simple application. And so your half-life is much more extended. And this is the rate limiting step from the skin patch. So there are other routes that are extravascular than oral. Oral very important, so that's why I focused on it, but I just want to give this overview. So after IV or arterial dosing, there is complete availability. With extravascular routes, this is not the case. It always involves a rate of absorption and an extent of absorption. So why do we apply the extravascular route? Well, often it's convenient, and systemic absorption may be desired. It's just convenient. In other situations, you actually want to target something locally. The target may be hard to reach, and systemic absorption may be undesired because it is associated with side effects. So the other routes are, for example, oral, intramuscular, subcutaneous, dermal, intraperitoneal, pulmonary, intrathecal, ocular, and the list goes on. But these are the most common ones. And I want to provide an example of intraperitoneal dosing. This is from my lab with an IP regimen for ovarian cancer. Bortezomib was investigational, carboplatin was standard of care. And as we can see here in the two concentration time profiles for bortezomib, the peritoneal fluid concentrations are much higher than plasma. Same thing for ultrafiltrable platinum, considered the active component of carboplatin therapy. Peritoneal fluid concentrations 1 to 2 orders of magnitude higher than in plasma. And so you're avoiding the systemic side effects, which you would get if you dose these drugs IV. And you get a much higher exposure locally where the tumor occurs, in the peritoneal cavity. So lastly, I want to discuss the bioequivalence. Bioequivalence is defined by the FDA as the absence of a significant difference in the rate and extent to which the active ingredient becomes available at the site of drug action. These kind of studies are done to establish a link, to show equivalence of different formulations, early, late clinical trial formulations, clinical trial versus stability formulations, and of course brand and generic versions. The test product is always the new formulation, the reference product is the prior formulation from which you have more data. So these are the situations. The test formulation can have a higher exposure than the reference formulation, and then the FDA would be concerned, and the company would be concerned about safety. If it's the other way around, you would worry about compromising efficacy. And if there's variability of your test formulation, then you would worry about both. So in general, the study design is such that you have these characteristics. It's very similar to the food effect study. I will not go through them in detail. And the sampling needs to be such that you capture the absorption, distribution, and elimination phase. You have 2 samples before the Tmax. You document at least 3 half-lives, number of samples, and oftentimes you sample plasma, serum, or blood. These are the PK parameters that are documented, and the statistics are very similar to what we've seen in the food effect study. So how do we come up with these differences that we find relevant, this 80 to 125% interval? Well, it was considered that 20% or less of an AUC or Cmax difference is not clinically significant. And so this 20% results in a drop to 80%, and in a log scale, the symmetrical distribution then results in 125% as the upper limit. So we express this 90% confidence interval in the log space of these geometric means of the test and the reference formulation, and these are 4 situations that can occur. If your formulations are equivalent and you have a reasonable variability, your confidence interval falls completely within these 80 to 125% limits. You can have a bioequivalence that is not exactly 100%, but if your confidence interval is really tight, it can still fall within the limits and pass the test. Your formulation on average can be exactly the same, but given high variability, the confidence interval can extend across your limits with high variability. And so what we can conclude is that bioequivalence means therapeutic equivalence, but bioinequivalence, like in the bottom situation, does not necessarily mean that your formulations are therapeutically inequivalent. If the sponsor has data that the dose-response curve is not too steep, then these exposure differences with 2 formulations may not result in clinically relevant effects. So the FDA guidance does allow for the waiving of these bioavailability or bioequivalence studies if a compound has been shown to be a BCS class 1 drug. And in that case, formulations can be tested against each other with the dissolution testing, where a compound passes the test if it rapidly dissolves for more than 85% within 30 minutes in less than 500 mils of each of these media. And so then you don't have to do a real in vivo human study. Thank you very much for your attention. I hope you found the lecture useful, and if you have any questions, please reach out to the program coordinator. Thank you.